Microbiology students, today I'm going to lecture over chapter 3, and this chapter covers observing microorganisms through a microscope. So we're going to talk about the compound light microscope, its parts, its uses. We're going to talk about other types of microscopy, and we're going to talk about staining microorganisms, specifically bacteria. And so with staining, you are applying a dye to those microbial cells so that you can better visualize those organisms underneath the microscope. So those are the topics of this chapter, and I hope you enjoy it. What you're looking at here is actually an image that was produced using an electron microscope. And these are bacterial cells. The species is Helicobacter pylori. This organism was first observed from the stomachs of cadavers in 1886. And it lives in the stomachs of humans today and is linked to causing ulcers. What you would observe using a microscope would be microbial shapes. Now this is, as I mentioned, an image produced with an electron microscope, so you're going to be able to produce really great detail, fine detail. But even with a compound light microscope, you would be able to see spiral shapes, or spherical or rod-shaped bacteria. This diagram is just showing you relative sizes of cells or organisms, as well as what types of microscopy would be used to visualize them. And a tick, for instance, you would be able to visualize a tick with an unaided eye. You would not need a microscope. However, if you wanted to look at red blood cells under the microscope, or to actually see red blood cells, you would need a compound light microscope. For viruses, they are going to be in the 20 to 60 nanometer size range, or 0.02 micrometer to 0.06 micrometer size range, and you would need an electron microscope to visualize a virus, either a transmission electron microscope or a scanning electron microscope. And we'll talk about these types of microscopy in this chapter, and we're also going to talk about light microscopes. So most of the organisms that we're going to talk about in this book are going to be in the 10 micrometer to 10 nanometer size range. Now, if you wanted to visualize the DNA double helix, you're going to need an atomic force microscope. These microscopes are really expensive, and of course at Rogers State we do not have an atomic force microscope. Microorganisms are going to be measured in micrometers and nanometers. And in any science course, you're going to be using the metric system, and it's based on units of 10. And so the prefix micro, we use the symbol mu to represent a micro, and micro means a millionth of a meter. Whereas nano, and we use the symbol N, is a billionth of a meter. And so 1 micrometer is going to be equal to 10 to the negative 6 meters. Okay, and that's because a micrometer is 1 millionth of a meter. So you can divide a meter into 1 million micrometers. You do need to be able to do metric conversions, especially among the units micro, milli, nano. You know, if you've learned a good method, stick to that method. And if you haven't learned a method, I will share my method. I'm a visual person, and so I like to use a small diagram to help me with the metric conversions. And so let's say this is just going to be your base unit. So this, let's say meter, and meter is your unit of, your metric unit of length. And so if we're going from large to small, so there's 2, so centi, deci, you have milli. We're really not going to deal with centi and deci. And then 2 more units and you have micro, 2 more and you have nano. And so let's say you have 100 nanometers, okay, 100 nanometers. And you want to convert that to micrometers. So you're going from a really small unit to a larger unit. So you're going to want to move your decimal place to the right. And so we're going to move our decimal place 1, 2, 3 spaces to the right. And if we do that, so we're going from, that's going to be 1,000 nanometers. So we're going to move it 1, 2, 3 spaces to the right. So that would be equal to 1 micrometers. And let's say we want to convert 0.001 micrometers to nanometers. And so now we're going from a larger, relatively larger metric unit to a much smaller. And so we're going to move our decimal place to the right. So now we're going from micro to nano. And so you simply just move your decimal place 3 spaces to the right to equal 1 nanometer. That's the method I like. If you had a really good teacher that showed you, or you just memorized how to do the conversions, that's perfectly fine. So if you just know that a micrometer is a millionth of a meter, a nanometer is a billionth of a meter, and a millimeter would be a thousandth of a meter. If you've always struggled with units of measurement and you want some one-on-one time with me to help you better understand, just shoot me an email and we'll set up a time to meet. You will be expected to do some metric conversions on the first exam. And they're going to be converting micro to milli to nano, and then knowing how they relate to the base unit. Now back to microscopes. We are going to talk about various types of microscopy, and in this image, this is showing you Leeuwenhoek's microscope, which I believe I said it had a magnification power of 300x. Leeuwenhoek's microscope would be most similar to just a simple hand lens, something maybe your grandmother would use to read the newspaper. So it's just one magnifying lens. Okay, so similar to a magnifying glass. His scope, Leeuwenhoek's scope, of course, had a way higher magnification power than your just hand lens. With light microscopy, you're going to be using visible light to observe the specimens. And visible light does have a longer wavelength than, let's say, electrons. If you're using electrons, then you would have electron microscopy. And so the types of light microscopy that we're going to talk about compound light microscopes, dark field, phase contrast, maybe briefly talk about differential interference contrast. We'll talk about fluorescent microscopy as well as confocal microscopy. Then we will talk about electron microscopes, both scanning and transmission electron microscopes or microscopy. This is showing you your basic compound light microscope, something you would find in a microbiology laboratory or another biology laboratory. Your illuminator is going to be your light source, so that's where the visible light is going to come from, and it's first going to pass through a diaphragm, and the diaphragm can control the amount of light that enters then the condenser. The diaphragm, you will have a little knob underneath the condenser, and you will adjust the light that enters depending on what magnification power you are using. With the condenser, it is going to consist of lenses that focus the light onto the specimen, which would be setting on the stage. And your objective lenses are going to contain magnifying lenses, and they're going to vary from 4x, 10x, 40, and then 100x. And they will rotate along this nose piece, so you can rotate among the different objective lenses. Light then, after the light passes through the specimen, it's going to be magnified by the objective lens. It gets magnified a second time via the ocular lens. Your ocular lenses typically have a magnification power of 10x. Now you can purchase different ocular lenses that have even a higher magnification power, but typically in the laboratory they're just going to be 10x. And this would be a binocular compound light microscope because you have 2 oculars. There are some that are monocular where you only have one ocular lens. They're difficult to use and they can cause a lot of strain on your eyes. But this is your basic compound microscope. You do have focusing knobs. So the coarse focusing knob is going to move the stage up and down. That's where, that's going to be the focusing knob you want to start using. And then once you have that specimen in focus, you then use the fine focus knob. In a compound light microscope, the image from the objective lens, as I mentioned, is magnified again by the ocular lens. And that's why we say compound, because you have 2 magnification lenses. And to determine the total magnification of that specimen, you multiply the magnification power of the objective lens by the magnification power of the ocular lens. And as I mentioned, the ocular lens is typically going to be 10 times, and we write that 10x whereas the objective lens, that magnification lens, may be 4x, 10x, 40x, or 100x. And so depending on which objective lens you are using, you can calculate the total magnification. So if you're using the 100x objective lens, you're going to multiply 100x by 10x, and that would be equal to 1,000 times, or 1,000x. And this is going to be the magnification power that's going to allow you to visualize the microorganisms that you would stain in a laboratory. And so it's important to know how to calculate total magnification and also know how to use the compound light microscope. I'm sure you'll watch a video within one of your laboratory activities on how to work the compound light microscope. Later in the semester, I plan to take my Winogradsky column up to the laboratory and see if I can't visualize the organisms that are growing in the various layers of my column. And hopefully I can use the camera on my Surface Pro to allow you as well to see what I'm looking at, and I will create a video of me using the compound light microscope just to better show you how to use one. And hopefully you've used one in the past. If you've taken cell biology, which you should have for this course, you've used a compound microscope. You may not have used a 100x objective, however. So this is just a diagram showing you the path of light within a compound light microscope. So you have your illuminator, so your source of visible light. It's going to pass through the diaphragm, which is going to control the amount of light that then enters the condenser. The condenser focuses the light onto the specimen, and as that light passes through the specimen, it then passes through the objective lenses, which have varying magnification powers. And then light is going to pass through a prism before it then passes through the ocular magnification lens. And so you would peer through the oculars, all right, to view the specimen that's on your microscope slide that you have situated onto the microscope stage. It's pretty straightforward. What's fun is that the fact that when the light passes through the prism, it gets inverted, and so when you move the stage, let's say forward, so you have a stage, adjustment knob. If you were to move the stage forward, whatever you were looking at in the microscope is going to move in the opposite direction. Okay, so if you move your stage to the right, that specimen is going to move to the left. And so skill using the compound light microscope really comes from practice, so hands-on practice using one. Now, another term that's important when we're talking about the compound light microscope is resolution. Resolution, or resolving power, is going to be the ability of those lenses to distinguish 2 points that are a specified distance apart, or the ability to reveal fine detail. So a microscope with a resolving power or resolution of 0.4 nanometers can distinguish 2 points that are at least 0.4 nanometers apart. With our compound light microscopes in the laboratory, they have a resolving power of around 0.2 micrometers. So your compound light microscopes are not going to have a resolving power of 0.4, maybe an electron microscope, but compound light microscopes a resolving power typically of around 0.2 micrometers. And just as a perspective, if you're talking, we talked about Leeuwenhoek's microscopes, and they were, more similar to a simple hand lens, but much higher magnification power. His hand microscopes had a resolution power of 1 micrometer. Okay, so of course the microscopes that you see in laboratories today have a much higher resolution than Leeuwenhoek's microscopes. And the best compound microscopes that you can find are going to have a magnification power of 2000x. As I mentioned, the microscopes that we have in our laboratory have a magnification power of up to 1000x. And the shorter the wavelength of light, that is going to provide for greater resolution. And that's why your electron microscopes are going to have a much greater resolution power or resolving power, and that's because those electrons are going to have a much shorter wavelength than visible light. To obtain a clear, finely detailed image using a microscope of a specimen, the specimen must contrast from the medium that it is suspended in. And the refractive index is the measure of the light bending ability of that medium. So the medium is going to be what the microbe is suspended in. It may be a nutrient broth, it may be a droplet of water. And so refraction is the bending of light as it passes from one medium to another. So refractive index is an important term to understand. And in order for us to contrast or make that microbe have a different refractive index than what it is suspended in, we stain the microbe with dyes. And that changes its refractive index so that its refractive index is different from that of the medium. And light may refract after passing through a specimen to an extent that does not pass through the objective lens. This is really important when we're talking about using the 100x objective lens, or it's also known as the oil immersion objective lens. And oil, we actually use a drop of oil when we're using that highest power objective lens because oil has the same refractive index of glass. And so this helps to prevent those light rays from being lost and not entering the objective. And I usually like to, in class, bring a glass stir rod, a bottle or a little beaker of water, and a beaker of oil. And you can do this at home if you have some glass at home and some maybe some just vegetable oil that you don't mind sticking a piece of glass into. If you were to place a glass stir rod into a beaker of oil, it would seem as though that glass stir rod disappears into that beaker of oil because glass has the same refractive index as that oil. Whereas if you were to place a glass stir rod into a beaker of water, you would still be able to see that glass stir rod because they have different refractive indexes. And so when we're using the oil immersion objective lens, this is the 100x objective lens, and that would allow us to achieve a magnification power of 1000x. You're going to place a small drop of oil on the specimen before then sliding the nose piece over to the oil immersion objective lens. And so that oil becomes part of the optics and will improve the resolving power. So let's start talking about the different types of light microscopy. We're going to talk about bright field illumination, and this is referring to just the compound light microscope. This is the type of microscope that you would use in a microbiology lab. And if you are viewing a specimen using bright field illumination, the specimen, they are going to appear as dark objects against a bright background. So light is reflected off the specimen and does not enter the objective lens. And this is the type of microscope that's most commonly used in laboratories. So with your bright field illumination using a compound light microscope, the microbes are going to be very small, they're going to be colorless, and you really can't see anything without staining them. And remember, by staining that organism with a dye, we are going to change its refractive index so that it's going to have a different refractive index than the medium that it's suspended in. Using bright field illumination, this is what the specimen would look like. And you would have a brightly lit field of view, and the specimen would appear darker. If you're viewing a protist, which is a larger organism, you could visualize some internal structures, maybe the nucleus, as well as if it's a plant cell, you could visualize the cell wall. This is a protist, and so it has this pellicle, which is an external covering outside of the plasma membrane. And this is the type of microscopy that you typically use in a microbiology laboratory in that teaching setting. However, if you're going to view bacteria, as I mentioned, they're much smaller than a protist, and you would need to stain them with a dye so that they would have a different refractive index than the medium that they are suspended in. With dark field microscopy, you are going to use a special condenser that will direct the light at an angle, and it's going to be the scattered light that enters the objective. And you're going to have then light objects that are visible against a dark background. Only light reflected off the specimen enters the objective lens. And dark field microscopy is going to be used to examine living organisms that are not seen using the bright field microscope. So they are below the limits of the size limits of the bright field microscope, or they're going to be organisms that do not stain or are distorted by staining. So living organisms is important. These organisms are going to be too small to be seen with the bright field illumination with your compound light microscope. And so you can use dark field microscopy to diagnose syphilis. So you would be able to observe Treponema pallidum, which is the pathogen that causes syphilis. However, you cannot visualize internal cell structures. And if you were looking at a specimen using dark field microscopy, the background or the field of view is going to be dark and the specimen is going to be brightly illuminated. Phase contrast microscopy uses a special diaphragm as well as a special objective lens, and it will allow you to examine living organisms and visualize those internal cell structures, or see the internal cell structures. So you could visualize cells undergoing division. You could see those chromosomes being separated from each other and move to opposite ends of the cell. And with phase contrast, you have direct light and diffracted light that are brought together. And what you end up seeing, no staining is required. You can visualize bacterial spores. You can see organelles if you were looking at a eukaryote. And what you're going to visualize then, it's going to be similar to dark field. So you will have a dark background, a brightly lit specimen. And so that direct light and diffracted light are brought together. And so the light areas are in phase. The gray or black areas are out of phase, hence phase contrast. Differential interference contrast microscopy is similar to phase contrast microscopy in that it uses differences in refractive index. However, with the DIC microscope, you're using two beams of light instead of one, and in addition to that, you're using a prism to split those light beams. It's going to allow you to give more contrast and color to the specimen. So similar to phase contrast, but with better resolution, the image is going to be brightly colored and will appear nearly 3D. A really important type of microscopy is fluorescence microscopy, and with this type of microscope you're going to use UV light, and UV light has a shorter wavelength than visible light, which would allow you to have better resolving power. And a substance that is fluorescent is able to absorb UV light and will then emit visible light. Visible light has a longer wavelength. You cannot see UV light, but you can see visible light. Some organisms are going to be naturally fluorescent. So there's some species within the genus Pseudomonas that are naturally fluorescent. So they're naturally going to absorb UV light and emit visible light that can be seen. You can use dyes that are known as fluorochromes, or fluorescent dyes that do naturally fluoresce. And there's various fluorochromes that are going to emit various colors of visible light, and there are, different fluorescent dyes or fluorochromes that are going to have different affinities for certain microorganisms. So some examples I have are, one example of a fluorochrome is auramine O, and it has an affinity for Mycobacterium tuberculosis that causes TB. And the visible light that is emitted is going to have a bright yellow color. Fluorescein is going to bind to Bacillus anthracis that causes anthrax, and the color of visible light that is emitted will have an apple green color. So just depending on the dye, that's going to determine the visible light or the color of that visible light that is emitted. And it also allows you to target specific microbes in a tissue sample. So in addition to just fluorescent microscopy, you also have fluorescent antibody techniques. These are also known as immunofluorescence or immunofluorescent techniques. And with this type of technique, you're going to use antibodies that have a fluorescent molecule that are attached to that antibody. So that antibody is going to bind to specific antigens on the surface of a specific microbe that you are targeting. So these antibodies are going to be produced in a laboratory setting, and they're going to specifically bind to antigens on the surface of a pathogen. And they do allow for rapid detection and identification of microbes in tissues as well as clinical samples. And so they would allow you to diagnose syphilis, which is caused by Treponema pallidum. Electron microscopes provide detailed images of cells, cell parts, and viruses, and they are using electrons instead of light. Electrons have a much shorter wavelength than visible light, which will greatly increase the resolving power. With electron microscopes, they're always going to produce a black and white image. However, they're often colored later for visual effects. So they're going to be used to visualize, objects or viruses and that are smaller than 2 nanometers. So you're going to have a resolving power of 2 nanometers. So used for images too small to be seen with light microscopes. Remember, light microscopes have a resolving power of about 0.2 micrometers. With an electron microscope, you're going to have a resolving power of 2 nanometers. So 2 nanometers is much smaller than 0.2 micrometers. 2 nanometers would be equivalent to 0.002 micrometers. The electrons are going to be absorbed, deflected, or transmitted based on the density of the structures in the specimen. And the practical limit, as I mentioned, of an electron microscope's resolution is 2 nanometers. And that would allow you to visualize viruses. There are 2 types of electron microscopes. You have transmission electron microscopes and scanning electron microscopes. With your transmission electron microscopes, they are going to produce a 2D image that is produced using a finely focused beam of electrons that pass through a specially prepared specimen. So you're taking ultra thin sections of that specimen, and that very thin section is placed on a copper mesh grid. And the electrons will pass through the specimen, then through a magnification objective lens, then onto a screen. With your transmission electron microscopes, you have a resolving power of 10 picometers. So that's even higher resolving power than the 2 nanometers that I mentioned. And you can magnify objects up to from 10,000 to 10 million times. Okay, so that's very high magnification powers and very high resolving power as well. One thing with the transmission electron microscope, you're going to have to kill the specimen, but it will allow you to visualize viruses, internal ultrastructure, and thin sections of cells. And as I mentioned, you do end up with a 2D image. And so this is an example of a 2D image that can be produced. And this is just showing you the beams of the electrons. So you're going to need you'll need a vacuum, you need magnets, you need electrons. As I mentioned, the transmission electron microscope, you get really high resolving power as well as really high magnification power. So this is just showing you another image produced using a transmission electron microscope. With scanning electron microscopes, again, you're using a finely focused beam of electrons, However, the specimen now is going to be covered with a thin coat of gold or some other metal, and when the electrons strike that specimen, you're going to knock loose showers of electrons that are captured by a detector. They are then going to be amplified, and you'll use a special screen to capture that image. And with scanning electron microscopes, you're going to produce a 3D image, and you will be able to visualize surface features of intact cells or intact viruses. So you don't have to take thin sections, so you can just visualize surface features. But however, you are going to have to kill that organism. And with your SEM or scanning electron microscopes, you can magnify objects up to 500,000 times their normal size, and you can achieve resolving powers of 10 nanometers. Surfaces of unsectioned objects. And as I mentioned, you're going to produce a 3D image. Okay, so with TEM, you get a 2D image. With SEM, you get a 3D image. And so this is showing you a paramecium that has been exposed, been visualized using a scanning electron microscope. So this is showing you some bacterial cells. Generally, you're going to produce a black and white image that is later colored for the effects. And now let's start talking about how you would prepare a smear for staining. The microorganisms, especially bacteria, if you're using a compound light microscope, they are going to appear colorless. Okay, so they're not going to be incredibly visible unless you stain them. And by staining them, you are changing their refractive index so that their refractive index is different from the medium that they're suspended in. And so staining refers to coloring microorganisms with a dye which will emphasize certain structures. And a smear is a thin film of material containing the microorganisms that has been spread over a slide. So you typically first start by preparing a smear. You then fix the smear onto a microscope slide, and then you stain. And so when we say microorganisms are fixed, we're talking about the fact that they are attached to the slide. So when you apply the stain and then rinse off the stain, you don't rinse off the organism as well. Heat fixing also, it attaches the organism to the slide. It also kills the microorganism. It will increase the stain absorption. So it will make those cells more readily able to take up the stain. And there's different ways to fix the organism onto the slide. You can use heat. You can also use alcohol. And so either one of those, you're going to denature surface proteins that allow them to stick to the slide. Now you can view live specimens using a compound microscope. You can also view unstained specimens. However, as I mentioned, they're going to have little contrast with the surrounding medium. But, if you want to study live specimens, you can study their behavior. And let's see if it's not going to play for me. So this is a video and it's not going to play. In this video, you would have seen a mixed culture of bacteria, and they would be moving about. And this is at a magnification power of 4000x. And so you can visualize live bacteria using a compound light microscope. However, if you can see them here even in the image, although I'm not showing you the video, they're colorless. Okay, so it's hard to look at cell shape, but you can study behavior. And now just to start talking about the stains that are used to add contrast to the bacteria. There are various types of stains. You have basic stains or dyes. You have basic dyes, acidic dyes, and these stains or dyes are going to consist of a positive and negative ion. Typically just one of those ions are going to have attached to it a colored molecule or a chromophore. And in a basic dye, that chromophore is going to be the cation. Remember, cations have a positive charge. In an acidic dye, the chromophore is going to be an anion. and that's a negative charge. So these stains are salts. Okay, you have in that molecule, you have a negative ion and you have a positive ion. Typically, only one of those ions are going to be colored or have a chromophore attached to it. With negative staining, you are staining the background instead of the bacterial cells. And with a negative stain, you're going to use an acidic dye. And to understand how the dye works, you have to understand the bacterial cell wall as well as the cytoplasm. The cell wall and the cytoplasm have an overall negative charge. So, with the cytoplasm, you're talking about like DNA has a negative charge, molecules on the cell wall will have a negative charge. And so if you apply a positive or basic dye to that bacterial cell, that dye is going to be attracted, since it's positively charged. Remember, a positive charge is attracted to a negative charge. So those positively charged basic dyes will have an affinity for the cell wall and will have an affinity for the cytoplasm. So they will attach to those structures and will stain them. They enter the cell, they stain the cell, they stain the cell wall as well. Some examples of positive dyes or basic dyes are crystal violet, methylene blue, malachite green, and safranin. Negative dyes, the chromophore is going to be negative, or it's going to be an anion, and a negative dye is going to be repelled by the bacterial cell. Okay, so a negative dye is not going to have an affinity for the bacterial cell. However, it will stick to the glass slide. And so with a negative dye, you're staining the background and the bacterial cell will appear colorless. There are different types of staining techniques. So a positive stain technique, you are going to use a basic dye. That's important here. And you will flood that prepared specimen with that basic dye. There are two types of positive staining techniques. You have the simple stain, and then you also have a differential stain. Both of these are considered positive staining techniques because you are using a positively charged basic dye. With a simple stain, you use a single basic dye. So you're just using methylene blue. And it does provide contrast. So you can observe the size of the bacterial cell, its shape, and its grouping or arrangement. Some bacterial species just occur as single cells. Some occur as clusters. Some will occur in chains. Or some just occur as two cells. So there's different groupings of bacteria. With a differential stain, you are going to use multiple stains, and the color of the organism will give you information on additional characteristics. So for differential stain, the most important differential stain is the gram stain, and it allows you to differentiate what we say are gram-positive organisms from gram-negative organisms. I am jumping ahead. I haven't, talked about gram-positive or gram-negative organisms, but you can differentiate those two large groups using a differential stain, specifically the gram stain. There are other types of differential stains. With the negative staining technique, you're going to use an acidic dye, and that acidic dye is repelled by the bacterial cell wall, repelled by the cytoplasm, and you end up with clear cells or opaque cells that are going to be visible against a dark background. There's no heat fixing that is required, and so you can observe cells in a more natural condition. Now, with the positive staining techniques, you are going to have to heat fix those organisms to the microscope slide. So simple stains, they use a single basic dye. You can highlight the entire microorganism to visualize cell shape as well as structures. If you use a mordant, okay, you can actually visualize the flagella. And a mordant is going to be used to hold the stain or to coat the specimen to enlarge it. So it's going to intensify the stain and increase the affinity of the stain for the specimen. So if you're going to try to visualize flagella, you're definitely going to use a mordant. You also use a mordant with the Gram stain. So it's going to better adhere that stain to the specimen. So two types of differential stains that we're going to talk about are the Gram stain, which I mentioned, and the acid-fast stain, which is used to diagnose TB as well as diagnose leprosy. So let's first talk about the Gram stain. And with the Gram stain, you're going to use two dyes. Crystal violet, which is known as your primary stain because it's the first stain that's used, and your safranin, which is a counter stain. And depending on the color of that organism after the staining procedure, that will allow you to determine if that organism is Gram positive or Gram negative. The steps in the Gram staining are, first you're going to apply your primary stain, which is with the Gram stain going to be crystal violet. It's a purple colored dye or stain, and you flood, you're first going to create a smear, heat fix the smear, and then you allow it to cool after heat fixing, and you apply the crystal violet. You allow that stain to set on the smear for about a minute, and then you rinse it. You then apply iodine, and iodine with the Gram stain is going to be the mordant, and the iodine will complex with the crystal violet. They form the CVI complex, so you get a crystal violet iodine complex. After you apply the iodine, you have that complex that forms, and all cells are going to appear purple at this point. Okay. The third step is the alcohol wash. Okay, so you're going to apply alcohol. That's after you rinse the iodine off. So any extra iodine that has not complexed with the crystal violet. And the iodine is going to, for the Gram-negative organisms, it is going to dissolve. They have a lipid outer covering, and it dissolves that lipid covering and makes those cells more permeable. And that crystal violet iodine complex can then exit the cell. So after this stage, those organisms that are Gram-negative, these are the rods, they're going to appear clear because you have rinsed out that crystal violet iodine complex. Whereas with the Gram-positive organisms, these are the spheres here. Gram-positive organisms have a thick cell wall, and the alcohol dehydrates that thick cell wall, essentially permanently trapping that crystal violet iodine complex within the cytoplasm. So the crystal violet iodine complex does not rinse out. And after this stage, they are going to appear purple. Now, the final stage of the Gram stain is to apply safranin. And this dye has a pink or red color to it, and it is going to stain all cells technically. Okay, but since the purple is a darker shade of color than the pink, you're not going to see the stain within the Gram-positive cells, but you will be able to see the Gram-negative bacteria. They are going to appear pink, whereas Gram-positive organisms will appear purple. So following these steps, you can differentiate organisms that are Gram-positive versus those that are Gram-negative. And it is early in the semester. We haven't talked about cell wall structure. This will make more sense after we cover Chapter 4. And after you complete the laboratory as well, that will help you to understand the Gram staining technique. So the Gram stain, it's going to allow you to separate two major groups of bacteria, those that are Gram-positive from those that are Gram-negative. So there's a large group of bacteria that have a particular cell wall structure. Those are Gram-positive organisms. Another major group of bacteria are the Gram-negative organisms, and they have a particular structure of cell wall. So Gram-positive bacteria, I mentioned they have a thick cell wall, and that cell wall is composed of peptidoglycan. So when you apply the alcohol, that that third step, it dehydrates the peptidoglycan in the cell wall, and that essentially traps those crystal violet iodine complexes within the cytoplasm, so they cannot leak out. With your gram-negative bacteria, they have a very thin layer of peptidoglycan, okay, and that very thin peptidoglycan layer is between two layers of lipids. So they have their inner plasma membrane, the thin outer peptidoglycan cell wall, and in addition to that, they have an outer membrane that's composed of lipopolysaccharides. And so the crystal violet iodine washes out because the alcohol essentially dissolves that outer layer of lipopolysaccharides. So it's a lipid, and so it gets dissolved. And when you dissolve that lipid layer, that crystal violet iodine complex can leak out. And so after you apply the alcohol, those cells are colorless, and then you then add the counter stain, which then stains them a pink or red color, depending on your perception of color. So the gram stain is going to provide valuable information, especially if you're thinking about the clinical setting. If you have a patient that has an infection, you can take a sample and use the gram stain to determine if the bacterium responsible for that infection is gram-positive or gram-negative. And that's going to allow you to choose a treatment. You know, there's some antibiotics that are effective against gram-negative, but aren't effective against gram-positive. And so you want to, make the right treatment option. Okay, so gram-negative are more resistant to many of your antibiotics because of their cell wall structure. And for instance, gram-negative are more sensitive to tetracycline. However, gram-negative are not sensitive to penicillin. So penicillin would be a drug that's effective against gram-positive, but not effective against gram-negative organisms. It'll help you make the right treatment option to treat a person. Another really important differential stain is the acid-fast stain. So the gram stain is a differential stain, and the acid-fast stain is a differential stain. This is an important stain, and it allows you to identify bacteria that are considered acid-fast. So you can identify the species of Mycobacterium that cause TB. So that would be Mycobacterium tuberculosis or Mycobacterium leprae. Mycobacterium leprae causes leprosy. Mycobacterium tuberculosis causes TB, tuberculosis. You can also identify Nocardia, which is another organism that is considered acid-fast. So this stain is considered differential because you can differentiate those that are acid-fast from many of the other bacterium that are considered non-acid-fast. And the stain that is used binds only to bacteria that have a waxy outer material outside of the peptidoglycan, but it is part of their cell wall. And this waxy material is called mycolic acid, and we'll talk about acid-fast organisms when we get to chapter 4 in more details. This is really important. So your mycobacterium or mycobacteria, they have this waxy mycolic acid as part of their cell wall. And two stains are used. You have your primary stain, which is called carbofuchsin, that's how you pronounce that, and it has a red color. And a counter stain that is used is methylene blue, and it is a blue stain. So those organisms that are acid-fast, they have the mycolic acid, they will retain this red dye after an acid-alcohol wash. Okay, those that are not acid-fast, they do not have the mycolic acid. That carbofuchsin will be rinsed out with the acid alcohol, and they will appear blue because of the counter stain. Okay, so let's look at the steps. So you have your primary stain, which is carbofuchsin, and it has a high affinity for that mycolic acid. You have to additionally use heat or or steam to drive that stain into the cell wall, and those cells are going to appear red. So all organisms after the primary stain will appear red. And then you're going to use a decolorizer. This is an acid alcohol. And the acid alcohol will not rinse the carbofucsin from the cells that are acid fast. Those that do not have the mycolic acid are in their cell wall, that carbofucsin is going to be rinsed out. And after the decolorizing step, they will appear colorless. So you then apply your counter stain, methylene blue. It is not going to penetrate that waxy outer layer, but it will penetrate the cells that are non-acid fast. So if the bacterium is non-acid fast, they are going to appear blue. If the bacterium is acid fast, they will appear red. So this is considered a differential stain. You're differentiating two major groups of bacteria, one that is acid fast and those that are non-acid fast. And so this is what a slide would look like after the acid fast staining procedure. And so these red cells are the mycobacterium. They do tend to clump together because of the waxy outer layer, and I don't believe there are any cells here present in the sample that would be considered non-acid fast. Okay, so just these red cells are acid fast. There are some staining techniques that you can use to distinguish parts of a microorganism. These aren't differential stains, they're considered special stains. So some microorganisms produce a capsule, and there's a particular stain that can be used to visualize a capsule. There are certain genera of bacteria that produce endospores, and there's a special staining technique to visualize endospores. And you can also use stains to visualize flagella or visualize the arrangement of flagella or the presence or absence of flagella. And these are special stains. With the capsule stain, a capsule is a gelatinous covering and it does not accept most dyes. So you would use a suspension of India ink or nigrosin. So these are negative dyes or acidic dyes, and they are going to allow you to contrast the background with the capsule, which the capsule is going to appear as a halo around the cells. And so these capsules, they are involved in virulence. Some organisms will produce capsules, but it's not associated with that organism's ability to cause disease or not. So it can be somewhat diagnostic to determine if you do have and so the capsule would appear as a clear halo. So this is the bacterial cell here, and the clear area around it is the capsule. That capsule is not going to accept the dye, but the bacterial cell will. So you can use special staining techniques to visualize capsules. Endospores are highly resistant structures that allow the bacterium to go into a dormant state. Let's say nutrients, an important nutrient is depleted, the organism will form an endospore. And we'll talk about endospores in great detail. They're really important. There are two genera of bacteria that produce endospores. Organisms within the genus Bacillus and organisms within the genus Clostridium. You will use a primary stain, which is malachite green. You are going to have to use steam or heat to drive that stain into the endospore. It's a structure that is resistant to radiation, resistant to high temperature, so it's a highly resistant structure. So you have to drive the stain into the protein coat of that endospore. You will then decolorize with water. So those that do not have an endospore, the malachite green is going to easily rinse out. And then you apply a counter stain that is safranin. And so after the staining technique, the spores, the endospores will appear green. So malachite green is green. And the vegetative cells are going to appear pink. Okay, or the mother cell will appear pink. And this is what your organism is going to look like after a endospore stain. So sometimes the endospores are still inside the mother cell. The mother cell is the cell that produces them. And sometimes they're going to be considered free spores. So if the mother cell lyses or disintegrates, it's going to release the spore. This is showing you, so the vegetative cells and the mother cells are going to appear red, and the spores are going to appear green. Okay, so that will allow you to visualize free spores or endospores. You can visualize a flagella, but you will need to use a mordant, and the mordant is going to coat the flagella, essentially make it appear thicker so that you can visualize it underneath the microscope. And so you use a mordant as well as carbofuchsin, and that will thicken the appearance of the flagella, making them visible under the light microscope. And some organisms may only have one flagella on one end of the cell. Others may have bipolar flagella. So for this particular organism, they have a tuft of flagella on each end of the cell. But you will need to use a mordant as well as a particular dye to thicken the appearance of the flagella. So it's a special staining technique. In the lab, you will complete an activity over the acid-fast stain, gram stain, and the endospore stain, but we do not do an activity over the flagella staining technique. Okay, so I hope you enjoyed the lecture.